As we welcome everyone, I see lots of great names here. So people, some of whom I've had the privilege of speaking to about the study, but we also have folks whose names maybe don't appear here because they're sitting in a room full of fellow residents, so we're happy to see that the coordinators have made that available to residents who maybe aren't able to access the actual webinar through their own devices. Great, lots of people joining. Thank you. So I'll just start quickly with an introduction. My name is Tara Kajax. I guess you could call me Dr. Kajax. I have a PhD in biomechanics, but what a privilege it's been to manage this project and to work with the coordinators that are working so hard at all of the homes and to have so many participants involved in the study, and of course the families supporting them and staff supporting their involvement. So super grateful for that, and super grateful for all of our partners who are part of this study. I want to welcome you to our first town hall of 2022. We've had previous town halls, but we recognize that we were recruiting up until the end of December for our retirement homes, and so this may actually be the first town hall that some of you have attended, so welcome. And for those that have been with us since the start of the study, which got off the ground just about a year ago, welcome back if you've already been to one of these town halls. Today we've invited Dr. Bowdish, one of our scientific leads, to present some of the current study results. Of course, we're constantly updating our results. We're doing it as quickly as we possibly can, so there's always lots of updates. And so Dawn will be sharing those results today, and then we have a few study updates that we'd also like to share with you. Dr. Bowdish is a Canadian immunologist and a professor in the Department of Medicine at McMaster University here in Ontario. She's a Tier 2 Research Canada Chair in Aging and Immunity, and she's also the Executive Director of the Firestone Institute for Respiratory Health at St. Joseph's Healthcare Center in Hamilton. She is half of the team that leads this. Dr. Andrew Costa makes up the other half of the scientific leadership on this project. Dr. Costa is the Schlegel Research Chair in Clinical Epidemiology and Aging and is an associate professor in the Department of Health Research Methods, Evidence, and Impact, as well as the Department of Medicine at McMaster University. He serves as the scientific director of the St. Joseph's Center for Integrated Care and the research director of the McMaster School of Medicine. He's also the Associate Scientific Director of the Canadian Longitudinal Study on Aging. His program of research makes use of health information to target, develop, and evaluate models of care in home and community care, emergency departments, hospitals, and of course, long-term care and retirement home settings. I will now share just a couple of housekeeping items, some of which I've already mentioned. But of course, upon entry to this webinar, we've muted all of your microphones. That's just to keep a little bit of silence and allow Dr. Bowdish to present. That said, we welcome questions and we're really excited to receive your questions. To do that, you can do one of two things. You can raise your hand within Teams, and if you don't know how to do that, we also welcome the good old wave, and we'll do our very best to watch for that. We've got Amraj sitting in the back watching for all of that, so we'll do our very best to watch for those waves and those hands up. And then, of course, you can also use the chat to share your questions. We will have a question and answer period at the end of the session, but sometimes your questions may be very relevant to what Dr. Bowdish is talking about, and we certainly welcome the interruption for those kinds of very relevant questions. I think that's about it for housekeeping, unless Dr. Bowdish, you have any questions? No? Okay, I'll hand it over to you now to jump into that presentation.
Thank you so much for the honor and privilege of getting to share our data with you, the people we care so much about. And it truly has been a highlight of my professional career, being able to work with the committed partners and research participants. And so I'm so happy to be here with you on behalf of my partner, Andrew Costa and I. So for those of you who haven't met us, I'll tell you a little bit about our story. So at the beginning of the COVID pandemic, both Andrew and I had longstanding research interests in older adults, and specifically those living in long-term care. Although we'd never met before, we knew of each other's work. I'd been doing studies on infectious disease and vaccines in long-term care and in older adults. And of course, he had been using evidence to improve care for older adults across the spectrum of the places we might end up where we age. And so when we, like all of us, were really moved by the incredible toll that this plague has taken in people in long-term care and retirement homes. And so we got together with this incredible leader of ours, Tara Kajaks, to build a research project understanding infections and immunity in the context of COVID. But also to use the data in a really quick and rapid way to inform policymakers to help make decisions about when to vaccinate, who should vaccinate, what kind of vaccines we should use in these communities. So I'm going to tell you a little bit about our story today, but ultimately we really believe that together partnerships like our collective partnership here can really improve the lives of many people living in long-term care throughout this terrible pandemic. So like I said, both of us were really deeply troubled by the incredible toll of deaths that happened in long-term care in that first wave. And so this is a graph where the gold bar or the gold numbers are the deaths in long-term care. And the blue is basically the age-matched community who's living outside of long-term care. And this incredible decimation of our community resulted in lost lives, yes, but also reduced opportunities in care and just, the sheer enjoyment of life for so many of the people who are living under these difficult conditions. Through an incredible collective effort with our partners, we were able to get, I think, 23 different homes now in retirement and long-term care involved in our study all over the province of Ontario. Currently, we have about 600, almost 700 people in long-term care who are in the study, and almost 332 who are in the retirement community. Effectively, what we've been measuring is immune responses in people over the course of the pandemic, so after their doses. And this is very much a textbook picture of what a dosing regime looks like in any vaccine. So you get your first dose, you have this little kind of wimpy peak of increase in antibodies or immune responses, and a second dose helps cement that and makes it a much more powerful response. But over time, that second dose will wane, and you need a third dose to really keep that immune system going. And in some of our vaccines, as I'll show you later, we'll have fourth doses. So today, what I'll primarily be showing you are antibody levels that people produce after these vaccines. And I'll be referring to things as the red, yellow, or green zone. So if you're in the green zone, that means you're sort of at peak immunity or at the equivalent of what a young, healthy person would look like. And the red zone will be under the level of detection, so no detectable antibodies, and yellow's intermediate, where we don't really understand where that level of protection really is. Right. This is a picture of the SARS-CoV-2 virus, the virus we have all grown to hate. And the little red bits that stick off of it are called the spike protein. Those spike proteins are what bind to our cells and allow it to get into us. And when we measure antibody levels, we measure two different kinds of antibodies. We measure antibodies that bind to that entire spike protein and sort of coat it. And then we measure antibodies to this really specific little tiny itsy bitsy domain called the receptor binding domain. And those antibodies, if we can get even one of them in there, what we'll see is that we can block that coronavirus from getting into to our own cells. And so I'll be showing you the antibodies that bind that entire spike protein. And when you see something called the RBD antibodies, it's a little itsy bitsy bit that that really binds to our cells. These are some of the data that we got after the second dose, and we were so pleased at how well these mRNA vaccines worked after the first and second doses in residents of long-term care. Historically, I'd done a lot of my previous research on the influenza vaccine. And in influenza vaccines, the ones that we're currently making, they don't actually work particularly well in older adults, and they don't always work in people who have complex medical conditions. So when the announcement came out in late 2020 that we were going to prioritize vaccinating, long-term care residents, I was really grateful because of this terrible loss of life and the burden of disease. But I was also concerned that they wouldn't be as effective. They surpassed all expectations. They reduced the number of deaths. They reduced the number of infections. It was wonderful. But what we quickly found out is that after a period of time, after the second dose, these antibody responses waned. So what happens, what you're looking at here is the blue bar is half the people. So the blue bar means half the people are above that and half the people are below that. You can see that even in 2 to 4 months after that dose, we had half the people were in that peak immunity, which was wonderful. A few people were in that red zone where they didn't have the immune response we were hoping for, and there were many in the intermediate. By 4 to 6 months, more and more people were leaving the green zone and entering the yellow and red. And by 6 to 8 months, we had fully half of our residents did not have detectable antibodies at this point. And I remember Andrew and we talked about these data one afternoon, and we were distraught. Actually, we were beside ourselves that we were concerned that fully half of our participants were unprotected. And so we talked about it, and the next morning we called each other, and I said, Andrew, I didn't sleep very well. And he said, I didn't sleep very well either. We have to let policymakers know that we might be looking at a place of our residents being really, really vulnerable. So in the next week, he and I went on what I call our speaking tour. We spoke to Public Health Ontario, the Public Health Agency of Canada, the Ontario Working Group for Advisory Council for Immunization. Frankly, anyone who was in a position to make decisions about this. We spoke to, we reached out to various public health units, and we started really advocating that a third dose was needed. This public speaking tour occurred in sort of late July, early August, and much to our delight, within less than a week, policymakers seeing this data, we were rolling out those third doses. And I'll show you an interesting little tidbit of information that we also were able to glean from these data. Of course, as I mentioned, there was a terrible toll of infections in the first wave or two. And one of the things we wanted to know is if we had to look into the future where we were living in this world where people's immunity was waning, the Delta variant was just creeping into Ontario, how vulnerable were our residents going to be? And so what we did is we looked at also the immune responses of people who previously had COVID in those early waves and people who'd never had COVID. And we did find that if we compared the people who had a previous infection in that sort of pink, I guess, peach, salmon, I don't know if you're a color aficionado, that pink color, and we compared those amounts of antibodies in the decline to the people who had the never been infected in sort of the orange, we saw that they had a little bit more protection and it lasted a little bit longer. So we were hopeful that even in a worst case scenario where we couldn't get third doses in time, there'd be a little bit more protection than in earlier waves, at least in people who'd had them. But we were absolutely, and as an immunologist, I was absolutely sure that more doses were going to be required. And in fact, 3 to 4 is always the magic number, almost always the magic number in immunology. When the immune system sees something that's brand new, believe it or not, it's more likely to forget it than it is to remember it. And the reason for that is you eat all sorts of new things that your immune system could respond to. You breathe, you touch. Your immune system does not get very excited about most of the things that you encounter. Otherwise, you'd be constantly inflamed. You'd constantly have that sick, feverish feeling that you get after a vaccination. So that first dose really sets the stage for the immune response, but without a second dose, you're almost certainly going to sort of forget it. Your immune system will forget it. So most of our vaccines require multiple shots. Measles, mumps, and rubella, which you would have had when you were, or possibly had when you were younger, is a 2-shotter, so it's the exception. But all our other vaccines, hepatitis, whooping cough, all these things have multiple doses included in them. Meningitis is actually an example where you can get 4 doses because you end up having your 3 doses, and then should there be an outbreak, oftentimes you get another one administered. So having multiple doses was not surprising, but where our advice to policymakers really was coming in is the timing. We wanted to make sure that we got the timing right to give people the optimal protection when we were expecting a new wave. So our public speaking tour that Andrew and I did, our knocking on doors of anyone who would listen, really was coming just at the right time. We were just starting to see an increase in infections in the Delta wave. We were just starting to see that curve increase again. But within a week, most of our partners had been contacted by various public health units in their vicinity. The 3rd doses came in, and then whoosh, those infections came right down. So for me, this was really one of the major wins of our study, was our ability to really mitigate the Delta wave. Now, let's look at what the data looked like after that 3rd dose. So I showed you already that 2, 4, and 6 to 8 month, where about half of our residents were unprotected. Now, some of our residents did decline getting the 3rd dose, and so we have not very many people, but we do have some people in that 8 to 10 month range. And again, you could see by this time, very few people are in that green zone of predicted protection, and the majority are in that red zone where we would worry that they would not be protected. But they get their 3rd dose, and then we have a nice big boost, and we have most of the people are up nice and high, at least till 4 months after their 3rd dose. I would like to draw your eye to one important feature of this graph, though. Even after this 3rd dose, you can see that there's a significant number of people who are in that vulnerable red zone. And long-term care is a special place because there's a lot of complexity going on there. There's lots of medications, there's lots of complex health conditions. As we get older, at least 1 in 5 of us, if not more, will have a condition that requires some degree of immune suppression. So high-dose steroids or drugs that treat conditions like arthritis can really suppress the immune system. We are currently trying to understand why some of our residents are unprotected from this vaccine, and I don't have any answers for you yet. But the important point to know is there is still a significant number of our friends and neighbors who are not protected. And these people are going to have to rely on the rest of us to do our part, to make sure that we are fully vaccinated, to make sure that all of us who are in contact with a vulnerable person are as vaccinated as we can possibly be, so we don't accidentally carry that into one of our vulnerable residents. Now, of course, we were so pleased with our work after the Delta wave. We were, it was such a nice, quiet time. So that's our little 3rd doses. That's where they begin. The curve goes down. We had these lovely few months where we had hardly any infections, and, the lab was ticking along and life was looking good. And then, of course, Omicron hit. And Omicron is a very different beast than previous versions of this virus. The Omicron variant has the majority of its mutations in that receptor binding domain part, that little itsy bitsy part of the spike protein that binds to our host cells. And as a result, we can't rely on antibodies that bind to that area anymore. They don't stick. They just don't stick to this virus. Instead, what we have to do is we have to rely on having so many antibodies, of having such a sort of a fresh vaccine that we are just absolutely covering every part of that spike protein that is not mutated with antibodies. And by making that spike protein big and bulky, it no longer fits onto that receptor and can get into our cell. So Omicron, because it's a very different virus than the one we were all vaccinated against, requires us to have quantity, vast amounts of these antibodies to stop us from getting infected. Is there a good way to get us those vast amount of antibodies? And we do think there is one knob we can turn to increase the amount of antibodies, and that is what type of vaccine we use. The data I'm showing you here are comparing people who had 2 doses of Pfizer, so P1, P2 is Pfizer 1, Pfizer 2, and then they got either another Pfizer, so Pfizer, 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 or they got Pfizer, Pfizer, Moderna, and then we break it down to people who had right after their 2 doses of Moderna, and then they go Moderna, 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 and we had only 2 people who did Moderna, Moderna, Pfizer. When we look at these antibody levels, what we see is that right before people got their 3rd dose, if you can compare, if you can draw your eyes to P1, T2, so the people who got 2 Pfizers, and now draw your eye to the M1, M2, the people who got 2 Modernas, you see that that blue line is a little bit higher in the people who had the 2 Modernas. Moderna has more mRNA in it, about 3 times more than Pfizer. Older adults and people who have complex medical conditions, people who are immunosuppressed, usually need an extra kick for their immune response. They need a bigger punch. They need more of an awakening, if you will, of the immune response. And increasing the dose is a proven way to do that. In fact, for influenza vaccinations, we actually have a specific high dose variant specifically for older adults, because for older adults, more is better. So even prior to that 3rd dose, our residents and participants who had 2 Modernas were already on average a little bit higher. Now, if we compare someone who's had 3 Pfizers to somebody who's had 3 Modernas, you can see that we have even more people up in that green zone and fewer people down in that red zone. So one of our recommendations for the 4th dose vaccine is that Moderna should be preferentially used for people in long-term care, retirement communities, and other places where older adults live. And in fact, based on Andrew and one of our many speaking tours, speaking to policymakers and sharing these data, which you have all so kindly helped us get, we recommended that older adults get the full dose of the Moderna for their subsequent vaccines, whereas any of sort of middle-aged people or young people would have had a half dose for their 3rd vaccination with Moderna. So our recommendation moving forward is to have as many of your doses be Moderna as possible if you're older. And currently, we're investigating whether this has helped protect against Omicron breakthrough infections. And here's just, and so we have a question about AstraZeneca. So very few of our residents, a few in the retirement homes, have had AstraZeneca. So everyone in long-term care was prioritized for the mRNA vaccines. A few of our retirement home members will have had AstraZeneca followed by an mRNA. The data is actually great. If you were one of those lucky middle-aged people who got an AstraZeneca first, followed by an mRNA vaccine, really wonderful immune responses and fewer breakthrough infections than people who got 2 Pfizers or whatever. So fear not, if you were an AstraZeneca. There was a lot of scandal at the time, but it actually looks like it's been a great investment in people's health. Now, I've just shown you some of the quality antibodies, and also when it comes down to quantity, we are still investigating this, but it does look as though Moderna actually increases the amount of all those total antibody numbers. We will be following, and I hope to report to you in a few months' time, well, maybe not a few weeks' time, if we find any relationship between the vaccine type used and the chance of breakthrough with Moderna infections or with SARS-CoV-2 infections. Now, there's been, obviously, we're focused on long-term care and retirement homes, and that's something that's my particular passion. But it's also important for us to keep the context of the general population in mind, because the majority of the infections that enter retirement homes and long-term care come from visitors and staff, people who enter the home. And so this graph is from the Ontario Science Table. It's a little bit hard to understand, so I'm going to walk you through it. If you follow that sort of purpley bar there, that was how well the vaccine was working. And in the Delta era, you know, you had your 2 doses, very few infections, looking good, looking good, looking good. Omicron hits, oh my goodness, 2 doses is not enough. People can start getting infections again. They don't die, they don't get crazy sick, but they can still get infected. We start giving the 3rd doses, and then we get an improvement in our vaccines. But there's a little gap here. We're not up to where we were in the pre-Omicron era. And a lot of that is because people are not rushing out to get their 3rd doses in the same way that they were rushing out to get their 2nd doses. We also saw that in this little dip where we saw an increased rate of hospitalizations was primarily in older adults who were in that period of waning immunity. So once they went and got their 3rd doses, life got a little bit better. And as we're getting those 3rd doses in, we're starting to see these lines between the number of infections in vaccinated and unvaccinated people really increase, where many of our infections currently are in unvaccinated people or people who've only had 1 or 2 doses. Let's look at our homes and see what's happening there. So from an immunology perspective, an asymptomatic infection is the best news you can give me. If you want to give me a Christmas present this year, give me an asymptomatic infection. Because what that means is you've got an infection, but your immune system has done what it's needed to do. It's fought off that infection. And now you probably have even a stronger immune response, because your immune system has seen this virus and seen other parts of it that are included in the vaccine. So now it's got even more things to attack if it ever sees this virus or a new variant again. One of the things I've been following with great interest is the number of infections in our homes, both the ones that a person was symptomatic enough that they got a PCR test, but also the ones where we can find evidence by way of antibodies to the virus that people didn't know they had. In our sort of calm before the Omicron storm period, there were, like I said, there were a few of these infections. You can see that little blip right in that Delta area when the 3rd doses came in. And then we also have a period where we saw some infections where there was never a PCR test done in a resident, but it does look like they developed some antibodies to the virus. Wonderful news in many ways, because that means that our vaccines were working. Then, of course, came Omicron, and we've had a number of PCR infections, but we will be following these people so closely, because what we want to know is their immune response stronger? Is it better? Is this likely to protect them against any variant that follows? And also to help our homes start to make decisions and planning about how they're going to deal with the next wave when it invariably comes. How many of the residents are going to be protected? What is that going to look like? In retirement homes, this is where our study started. So we started looking for antibodies, and there were fewer infections to date, fewer PCR confirmed ones, and a little bit of these antibody ones, which we think were asymptomatic. So great news. Asymptomatic infections are the best gift you can give right now to me as an immunologist. Now, I always feel like, you know, I'm kind of like a harbinger of bad news. I'm kind of like the Debbie Downer that nobody wants at a party, because I'm always talking about infections. So I hope you will allow me to relish and share with you a good news story that our studies found. I've been talking a lot about antibodies. Antibodies are the easiest part of an immune response to measure. We can measure them in the liquid part of the blood, and we can do all these fancy things that I've been telling you about. Our current understanding, though, is complete protection requires 2 arms of the immune response. Your antibodies stop you getting infected in the first place. And they also probably help a lot with transmission, because if they're binding and covering that virus, even if you spit it or cough it or share it, if they're all covered up with those antibodies, it probably can't infect the next person. So antibodies are really important for getting infected and then for sharing the infection. But one of the things that's been very different about this wave is that we've seen a huge improvement in vaccinated people in hospitalizations and death. The severity of the illness in many cases has been less. And we think that's because these vaccines have stimulated what we call cellular immunity, or the white blood cells. These do slightly different things than antibodies, and we think that that protection is helping people not go to the hospital and not die. The other wonderful bit of news about cellular immune responses is they seem to be more conserved across variants. So Omicron did not develop a bunch of mutations that allowed it to escape cellular immunity. And we think this is nothing but good news. And in fact, cellular immunity is incredibly hard to measure. It takes days, actually it takes 4 days and 3 people, believe it or not, in the lab to do these experiments. But when we've looked at our residents, we see that very few of them are in that unprotected zone for cellular immunity. Most of them, after 3 doses, are able to make those cells that will go and attack those virus-infected cells. So we're very excited about that. And we're also seeing that this looks a little bit better with some more Moderna doses, further justifying our recommendation that Moderna should be the vaccine of choice. So we hope that this is important information to share with vaccine makers who are trying to generate new vaccines that target the cellular immunity. And it's also good news for understanding how people will deal with this in the future after they've been vaccinated, if their immune cells can take up the fight. So again, when I show you these graphs here, we saw, people who had their 2 doses, yes, they were getting sick, but we didn't see such a big dip in hospitalization and deaths. And we think that's because the cells were doing their job and stepping up when the antibodies just didn't work anymore. Now I want to draw your eye to this picture here. And this picture here is, when I saw it, I had a bit of an emotional reaction. Early on, those gold lines are the deaths in long-term care. And the blue are sort of age-matched people in the general population. And what you could see in the first 2 waves, yes, we had this terrible loss of life. We had this period where we were doing so many things. There's so much isolation, so many rules to keep people protected. We started getting them vaccinated. Omicron came. We started to have problems again. But if you look at the very tail end of that graph, if you look on the very right-hand side, what you're actually seeing is that after those 4th doses, deaths are lower in long-term care than they are at people living in the community of the same age, many of whom have not had a 4th dose. So being really proactive about recommending those 4th doses and getting those 4th doses into the population as Omicron was hitting looked like it was effective in mitigating some of the terrible human cost of this horrible disease. But we're not done. We're not done. We haven't figured this out yet. I have not cured Omicron, have I? So what we're going to do next is try to include an additional sample collection. Like I said, those antibodies seem to be really important in stopping transmission and blocking the infection. And there's antibodies in your blood, yes, but that's not where Omicron infects you, is it? It infects you in your nose, in your lungs, respiratory tract. There is considerable debate in the immunology community right now if our vaccines are giving us enough antibodies in our lungs, in our nose. And an easy, quote unquote, way of measuring these antibodies is by looking at them in spit. So your mouth has lots of these antibodies in them. And what we're requesting to do for our residents who are willing and able to give us saliva samples to spit in a tube when we take their blood next. And we're going to see if maybe we can explain why some people don't seem to get this infection and other people do because of the levels of these antibodies in their blood. Are they there waiting to coat this virus so it could stop an infection? And if not, why not? We also are monitoring this very closely. We've been able, and I've been so impressed by policymakers and how receptive they are to listening to us and taking our recommendations and trying to do everything we can. But we need to know some things. We need to know how long do we have from protection during this Omicron wave. We need to know what vaccines are the right vaccines. Should we be going with Moderna for the rest of our time? Our residents who've had 3 Pfizers, do they need a little Moderna top up? We need to be able to make some good decisions about making sure people get the right vaccine. And we still don't understand why some people aren't protected and some people aren't protected. So currently, we are investigating all our residents and participants who got Omicron infections and saying, what's different about them? Did they not have the right cells? Did they not have enough antibodies? Did they not have the right antibodies? And these are really important because in the future, when we design vaccines better, if we know what we're aiming for, if we know what the bar is, then we can aim to get to that bar. But currently, we don't know what the bar is. I couldn't tell you honestly how many T cells you need to be protected, or do you need antibodies in your saliva or not. But I think with the samples that we are currently collecting and plan to collect, we'll be able to get those answers. And I just need to have a huge thank you to our incredible team. I mean, our participants have just been a joy to get to know. And I'm always so thrilled to hear their stories about what they like about the study. This has been the biggest study I've ever had the pleasure of being involved in. And we've had such incredible commitment from management and frankly from our research coordinators and so many of our homes who've done such an incredible job in recruiting and sharing with our participants and getting us all those samples, even in extremely difficult times. And then you would not believe how many people touch those blood samples when they get to the lab. We have our incredible manager, Tara Kajax, who organizes with our logistics team and our quality assurance immunology team, who makes sure that everything's processed correctly. And then we have what I call the brain trust, who comes and who works on these cells, and not a drop of blood is wasted. We have thousands of samples in our freezers and our fridges right now, and we're learning from them absolutely every day. And it's a pleasure and an honor to be able to share these results with you. And I thank you again from the bottom of my heart for your participation.